Good afternoon, and welcome to the Alamo Breast Cancer Advocate Chat. My name is Mary Jimenez, and I'm the director of the Alamo Advocate Program and chair of the UTMA's Community Outreach and Advisory Board. It gives me great pleasure to introduce our speaker for today, Dr. Lewis. For more than 30 years, Dr. Lewis has headed a tumor cell biology preclinical drug development lab in Genentech research with a focus on therapeutic antibodies and antibody drug conjugates. She and her team investigate target selection and target biology with the goal of understanding the optimal large molecule modality for each target and indication. Her early contributions in studying HER2 receptor tyrosine kinase was important for the development of Perjeta, Herceptin, and Kadcyla. She has continued her efforts in ADC pathways with a focus on diverse targets across both solid tumors and hematologic malignancies, working with an expanded arsenal of novel linker drugs, including more targeted non-chemotherapy ADC payloads. In recent years, Dr. Lewis has been involved in the development and FDA approval of inavolisib, a new medicine for breast cancer. Dr. Lewis received her undergraduate degree in microbiology at the University of Texas at Austin and performed her graduate work at UT Southwestern Medical Center. She joined Genentech after graduate school and has been with Genentech for over 40 years. Her current role is Distinguished Scientist in the Discovery Oncology Department. She has authored over 80 publications and received the Distinguished Alum Award and inducted into the Hall of Honor at the University of Texas at Austin's College of Natural Sciences. Please join me in welcoming Dr. Lewis as she will be discussing From Bench to Bedside, Understanding ADCs and What the Future Holds. Dr. Lewis?
Thank you, Mary, for such a kind introduction. Mary and I have known each other for years, so it's really good to see you again and to be able to participate in this Alamo Advocate Chat. Saying how long I've been at Genentech dates me. I will say I just celebrated my 40-year anniversary, so it hasn't been that much over 40 years. And I do want to say as a disclaimer, I'm a full-time employee of Genentech, and some of the things that I may talk about in terms of opinions are those of my own and not necessarily the point of view of the company.
Wonderful. So great. So Dr. Lewis, before we get started, could you please share a little bit about the HER2 positive breast cancer evolution and how it evolved, how you got involved in the drug development in this field, starting with your early work on Herceptin?
Yeah, certainly. So I feel very, very lucky and very fortunate that shortly after I started working, actually the year I started working at Genentech, the HER2 gene was cloned. And shortly after that, our antibody group was making antibodies against the HER2 receptor, really for us to just study in the lab. At that time, there really wasn't probably a therapeutic goal in mind. But as we studied more, we very quickly understood that antibodies against HER2 could block the function of this receptor. This is a very potent oncogene, an oncogenic receptor that drives cancer cell growth in cells that overexpress HER2. And we made the observation that some of these antibodies, and one in particular, which actually became trastuzumab or Herceptin, was very good at blocking the growth of HER2 overexpressing cancer cells in culture. And I had not worked on cancer cells before I got to Genentech. I did my graduate work in inflammation. And so this was all very new to me, but it was very, very exciting and fun and rewarding to see these sort of basic discoveries in the lab actually start really meaning something and driving the advancement of a potential therapy program.
Yeah, so definitely it's certainly a place. Now we look at what our market has today, so many HER2 positive therapies, certainly bad players turned into good opportunities for patients to now have options for treatment. So why do you continue to work on and try to improve drugs for HER2 positive breast cancer?
Yeah, no, that's a really good question. So, after our work led to the development and approval of trastuzumab, Herceptin. We had other antibodies that were in this small arsenal that we had originally started with, and we were using them to really dissect deeper into the biology of the HER2 receptor. And it's a complex system. It's a complex system of different receptors and different activating growth factors. And we discovered that there was a second antibody that could block signaling in HER2 positive cells that worked very differently than trastuzumab did. And so we did a lot of studies to understand how this worked and why it worked and why it was different than trastuzumab. And that antibody was the one that became pertuzumab or Perjeta. This antibody had a very difficult development life because our original findings showed that its mechanism was so different than trastuzumab because it blocks growth factor signaling through HER2, whereas trastuzumab works a little bit differently. And that it could actually work in the context of non-HER2 amplified models, preclinical models. We looked at breast, ovarian, prostate. And so in the early days of pertuzumab, because we were developing trastuzumab, pertuzumab actually went into early clinical development in non-HER2 amplified indications, and nothing really worked very well. There was like little hints of activity, but going back to the drawing board, I think we really finally realized, especially working with collaborators, that we get the most bang for our buck if we focused on the HER2 amplified setting with pertuzumab and combining it with trastuzumab and combining those multiple mechanisms of action together to really drive like deeper, tumor growth inhibition. So this was a very long journey, but I think as people know that use these in the clinic, that this combination has shown clinical benefit.
Yeah. So many different pathways, right? And so turning off one isn't enough, right? And I think that's the story, to keep looking at more pathways. So speaking of other pathways, what additional ADCs have you worked on and are continuing to work on?
Yeah, so the first ADC I worked on is Kadcyla or TDM1, and I will say that we started our work. So ADCs are antibody drug conjugates. I think everybody knows this modality where a drug is conjugated to a tumor selective antibody for delivery, to hopefully tumor cells. We started our work on ADCs in about 1999, so it's been well over 25 years now. And part of working on a HER2 ADC was Herceptin had just gotten approved. We were excited about the clinical data, but we also thought that maybe we could develop something that worked better. And knowing that Herceptin is approved in combination with chemotherapy, the idea was actually to combine the Herceptin antibody and a chemotype agent into one molecule. And that was the first ADC we worked on, which was Kadcyla. And that was approved in 2013, has been approved in 2 different indications. But it's really interesting because as the ADC field has evolved, so have the advances and the technologies and the findings. And now we know that our early design of Kadcyla, for us, it was good because it was a very well tolerated and efficacious ADC. But some of the design elements also had limitations that have been overcome by some of the newer ADCs that can now target HER2 positive breast cancer in a much more sort of diverse and heterogeneous HER2 expressing patient population. So this field has really exploded. It is a very competitive area, and so much of the ADCs work that we have been doing have been to move away from the HER2 field and look at the other types of breast cancer. So we know that there are 3 subtypes of breast cancer. There's HER2 positive, there's triple negative breast cancer, and there's hormone receptor positive breast cancer. These other subtypes of breast cancer are continue to be in need of therapies. There are now a couple of ADCs that are approved in either triple negative breast cancer and or hormone receptor positive breast cancer. But they are used to treat patients in the later lines of setting, especially in hormone receptor positive breast cancer, where chemotherapy, and these are like chemotherapy drugs, are not given until the later lines of setting because there are, of course, really good drugs to treat the earlier lines. But there are additional targets I think that we can tap into, and certainly additional types of linker drug technology, the different types of payloads, which is the drug on the ADC, that I think we can tap into to try to make advances in these other two subtypes of breast cancer, like we sort of have already done in HER2 positive breast cancer.
So like in the triple negative breast cancer, hormone positive breast
Yes,
cancer.
exactly.
Okay, yeah, that's really critical. There's a lot of information coming forward at San Antonio Breast, still covering these pathways. So this is very, very helpful for our advocates listening today as they learn more about these treatment options for, I think what used to be when you and I worked together, a very aggressive form of cancer with limited options. So very, very hopeful. So thank you for sharing. Is there anything additional that you'd like to mention, Dr. Lewis, that we haven't covered today on the future of ADCs or future of breast cancer in general that your lab is working on? This always gives our audience today hope on what's on the horizon.
Yeah. So yes, so I think everybody knows that the ADC field is kind of crazy now. I mean, there are so many different ADCs that are in preclinical development. There are so many that are in clinical development. It's really it has exploded over the last probably 5 or 6 years, because we are now seeing areas not only in breast cancer but many other types of cancer as well, where ADCs work, are getting approvals, and this has led to sort of a blossoming and an evolution of work on ADCs. Now, that being said, having been involved in ADC work really from the get-go, like I say, more than 25 years ago, back then, our idea really was what everybody was thinking about ADCs, and that you have an antibody. So like, for instance, we use trastuzumab to target HER2. You have an antibody that's very selective for a tumor target, and you link a potent chemotherapy type payload to that, and you let the antibody deliver that chemotherapy drug to the tumor. That was the goal, and the sort of the sort of idealistic hypothesis around that. In reality, it has turned out that ADCs are still fairly toxic drugs, for mechanisms that we didn't appreciate so long ago. So there is tumor selective delivery of these potent payloads, but you can demonstrate ADC uptake in a non-target or non-specific way into tissues, normal tissues all throughout the body, which is basically what leads to the toxicity of ADCs. Many of the ADCs that have been studied, their toxicities by and large are not on target, but they're what we call off target, meaning they're delivering the drug through nonspecific uptake into tissues, and that drug is getting in and damaging the tissues. This, of course, is dependent on the type of linker, and the more cleavable the linker is, the more this can happen in a way, and the mechanism of action of the payload. And certainly some of the newer payloads, which act on DNA, versus the old generation of payloads, which acted on microtubules to disrupt cell division. These newer payloads tend to have sort of deeper activity and yet sort of more severe toxicities. And so I think this is the big challenge, and certainly for me, and I'm speaking about my opinion now, having been in this field for so long, the big challenge for me now is for ADCs to figure out a way to have them be as efficacious as this newer generation of ADCs is, without the associated side effects and toxicities. And there are a lot of different people working on this, and there's a lot of different ways you can approach this, from engineering the antibody with different properties, to engineering the linker, to coming up with different types of payloads to conjugate to the antibody to sort of try to get around this. It will be very challenging, but I think this, to me, is sort of the next step of what I would like to see happen. And along those lines, the focus of my lab over the last probably 6 or 7 years has been to try to find really unique and targeted payloads for ADCs that are not chemotherapy drugs. So to date, every approved ADC has some version of a chemotherapy type drug that is conjugated to the different antibodies that recognize the different targets. And so what our idea is to look at intracellular pathways in tumor cells that tumor cells may be very dependent on for survival, that really drive their survival, that you might be able to target with a small molecule that only targets that pathway and not pan-essential pathways like DNA or like targeting the microtubules in the cells. So this is what we've been working on. It's very challenging because these payloads typically aren't as potent as chemotherapy type payloads. But if you have the right target and you know the optimal amount of a drug to put on the antibody and all the other associated things that go along with developing ADCs, I think eventually there will be success in this area. We're not the only ones working on this. There are other companies working on this as well. And I think this will be maybe the next wave of ADCs because it's going to take a while to figure this out. But I'm hopeful if we can figure out these pathways to target in cancer cells that are where cancer cells are very uniquely dependent on these pathways versus, say, normal tissue, that at some point, hopefully in my lifetime, we can come up with a generation of ADCs that do work really well and have clinical activity and maybe less of the associated toxicities as the current ADCs do.
That would be wonderful. You know, it's hard. You want to leave the chemotherapy behind, or find a way to deliver a chemotherapy in a safe way where it goes right into where it needs to go and not affect everything else and the good cells. Yeah, so it makes a lot of sense. Thank you so much. Very, very helpful. Always been an amazing advocate and scientist for breast cancer, and we appreciate your years of contribution to research. On behalf of the Alamo Advocate Program, thank you for joining us today, Dr. Lewis, and a special thank you to all our breast cancer survivors and patients that continue to participate in clinical trials. Thank you.
Thank you very much, Mary.